So this is our clicker review from today. A lot of people asked me if I would please make a recording of this, particularly about the Hardy-Weinberg stuff, because that is what your short answer is going to be about. So I'm going to run through the other stuff fairly quickly and spend a little more time on that. So the first one shows a diagram, and they want to know what kind of structures those are. So if you look at that, this is sort of a classic picture of homologous structures. Homologous structures, they are structures that are similar in their structure, underlying bone structure, for example. For example, we might highlight this bone and see that even though these organisms are all different from each other, they all have this underlying bone. It may be longer, shorter, fatter, whatever. Vestigial structures would be more like the appendix, a hip bone in a whale where whales don't use it for anything. Analogous structure is oftentimes going to be there to trip you up. And analogous structures are those that are not related in an evolutionary sense, but maybe they developed because the organisms were under the same pressures. So like the wing of a bird and the wing of the butterfly both allow them to fly, but they're not related to each other. It's not like the wing of a butterfly would have evolved into the wing of a bird. Their underlying structures are completely different. So homologous structures for this. Next, two organisms showing similar structures in their hemoglobin. The clue here is that this is a chemical. If they mention hemoglobin, insulin, anything that you recognize as a chemical or on the chemical level, or they say their DNA is the same or their amino acid sequences are the same, then we're looking at comparative biochemistry. The keywords for embryology are going to have to do with before birth, and anatomy is always going to deal with either homologous or vestigial structures. The sudden disappearance of dinosaurs and the sudden appearance of many mammals. What they're going for here is the idea that you had this period of time from like 250 million years ago to like 65 million years ago, and we don't see a lot of changes. Everything's kind of stable. All of a sudden, 65 million years ago, all the dinosaurs are gone, and all these mammals appear. So what we're seeing is punctuated equilibrium. We're seeing periods of stability and then these spurts of change. And that's what they're going for there. The opposite of that would be Darwin's theory, which would have been gradualism, saying that the changes would have been slow and steady. You know, that we see this type of dinosaur, then this type of dinosaur, then this type, and then the alligator of today. And it's something that was slow and steady. Okay, so let's spend a little time on this one. So 64% of this deer population shows a particular dominant trait. So if they say that it shows a trait, then they're definitely giving you something along the lines of p squared 2pq. I'm going to move the follow-up question over here. They're giving you something along the lines of p squared 2pq q squared, because this has to do with organisms showing traits, whereas these are allele frequencies, and they have to do with how common, let's say, capital A is versus lowercase a in the population. So if you say something about genes or alleles, they're talking about P or Q. The only piece of information that would be completely useless to give you, if the only piece of information you had was 2pq, you couldn't solve the problem because you have two variables. But if you've got p squared or q squared or p or q, you can get any other piece of information you want. So if 64% shows the dominant trait, this is a little bit tricky because showing the dominant trait would include everybody that's big B, and everybody that's big B, little b. Which means this 64% is representing p squared and 2pq, which means we can't solve this problem with this information directly. But if we know 64% show the dominant trait, that means the other 36% show the recessive trait. And we know that the recessive trait, little b, is q squared. So if q squared is 0.36, Q is going to simply be the square root of that, which is going to be 0.6, and then P plus Q equals 1, so that means P is going to be 0.4. So the answer here is B. Notice how easy it is to figure this out once you know what piece of information you're being given and what piece of information they're asking for. Then it's very just simple math. All right, let's take a look at the follow-up question here. Okay, so in our follow-up question, we say the population consists of 200 deer, and how many deer would be homozygous dominant. So if P is 0.4, we know that P squared represents our frequency of homozygous dominant, our percentage. So 0.4 squared is going to be 0.16. And then 0.16 is going to be our frequency of dominant, but we have 200 deer, so to get how many deer would be homozygous dominant, we would multiply 200 times 0.16, and if we do that, we get, I believe, 32 deer. Okay, and so that's telling me how many deer are homozygous dominant. The actual question here is how many deer should show the dominant trait. So what they're really asking you for here is P squared plus 2PQ. Now technically you could kind of do a trick here, because if 64% show the dominant trait, then technically 0.64 times my 200 deer should give me how many show the dominant trait, which would be 128. You also already know, if you didn't think about doing it that way, you also already know that 36%, 0.36, we said was Q squared. I had that from the original question. So if Q squared is 0.36, that would mean 0.36 times 200 would give us our recessive deer, which would be 72. So we would have 72 that are recessive. If you didn't remember how to do both of these, it doesn't really matter, because once you know how many are recessive, you can subtract to get how many are dominant, because you know they have to add up to a total of 200 deer. So there's several different ways really that you could solve this. You could get P squared and get 2PQ and add them all up, or not even necessary. Like I said, you already have this number in this particular case. You could use this number, or you already calculated this number, so you could get this one and then subtract from 200. All right, so now we know how many deer are dominant, how many deer are recessive. Our next follow-up question, I'm going to move this out of the way and bring this over here. So follow-up number 2, 50 years later, there are now 400 deer. 230 show the dominant trait. Is this still in equilibrium or not? So let me squash this into the corner. All right, and then I'm asking you to do a chi-square test to see if indeed it does fit into, that it's still in equilibrium. All right, so we have 400 deer, and this time 230 of them show the dominant trait. Well, the first thing we need to do is we need to say, okay, so we have 400 deer. We have a certain number of expected, we have observed, and then remember we get our chi-square. So out of 400 total deer, we know that 0.64% of them, 0.16, are going to, should be dominant, and then 0.36 should be recessive. So if we take our 400 and multiply it times 0.64, that should give us how many should be dominant, and if we multiply it by 0.36 or subtract or whatever, that's going to give us how many should be recessive. So when I worked this out, I found that my expected was 256 for our dominant and 144 for our recessive. Technically, you could also just double these numbers that you got here, because it's double the population size. They now give us observed. So they say that there were 230 that actually showed the dominant trait. We can subtract, and we would know that 170 therefore must have showed the recessive trait, because these add up to 400. If we then plug this into our chi-square, I'm not going to spend too much time on this because you're already supposed to know how to do this, but it would basically be 256 minus 230 squared over 256. And then for our other one, it's going to be 144 minus 170 squared over 144. If we plug these in, let's see, I wrote this down. I believe we get 2.64 for this one, and then we get 4.69 for this one. And if we add this up, we get 7.33. You have 1 degree of freedom. If you go to the chi-square table, you're going to see that our cutoff is 3.86. Since this number is bigger than our cutoff, we would reject. We would say that this population is not in Hardy-Weinberg equilibrium. Something must have changed. So that's how you would do this problem. All right, moving on to some of the other questions. Some of these are just fact questions. Which of these would have led to the development of species of honeycreepers in the Hawaiian Islands? So your keywords here, definition, simple definition, lots of species are developing and it's happening in an island chain. That is a clear definition of adaptive radiation. What has to happen in adaptive radiation is we see they get separated on different islands. There's an ancestral species, but there's already variation. When they get separated, they're no longer coming in contact with each other. There's different selective pressures because some may settle, for example, on the beach, some settle in the rocks, some settle in the forest. So the pressures on them are different. So maybe having a longer beak on the beach is better. Having a darker color in the forest is better. And so natural selection is going to play a role here. But not just natural selection. You're also going to see these are small populations. So we may see some genetic drift playing a role here. We also would likely see a few mutations thrown in there. And so over time, with all of these different things going on, we end up with these multiple species. So some people, when we did this in class, chose genetic drift, and genetic drift does play a role. You're talking about small populations being separated from each other, but adaptive radiation really is just definition spot on what this question is asking. Sterile hybrid animals shown below are examples of different, I'm not going to read all the choices. Basically, we went over this in class. Hybrids, if two organisms cannot mate and produce fertile offspring, they're considered different species. Now there's different levels of this. They could have different courtship rituals. They could mate at different times of the year. They could physically not be able to match up their sex organs to make an offspring. In this case, they can make an offspring. The donkey and the horse make a mule. You get the Z-donk here. This is the comma. He's so cute. And then the liger. These are all, but the problem is these are sterile organisms. I think female ligers are actually fertile, but the males are sterile. So you couldn't have a population of ligers that could then reproduce with each other and keep the thing going. So this would be post-mating isolation. Sometimes you'll also see, I think in the PowerPoint, I used post-zygotic isolation, meaning after this, there's a problem after the zygote forms. A zygote can form, but at some point after that, we still don't see their gene pools mixing. If this female prefers the male because he can balance on one leg, and this is a genetic trait, we will see sexual selection or non-random mating. And therefore, the females pick these males, and over time, because these males pass on their genetic trait to more offspring, we see a shift in Hardy-Weinberg equilibrium. This would probably be directional selection. Be careful with sexual dimorphism. That just means when the males and females are different colors. For example, these ducks actually exhibit sexual dimorphism. The males are colorful, the females are ugly. But that's on purpose, so they can guard their eggs and be camouflaged.